Greetings and welcome, everyone. My name is Lokita Maka. Thank you for joining me in this session. I want us to discuss bacteria. For the contents of our session, we're going to discuss prokaryotes, identifying prokaryotes, their energy, respiration, reproduction, as well as their relationships, that is the symbiosis. All living things or all living organisms are classified into three major domains. That is the domain Eukaryota, which includes the eukaryotes, domain Eubacteria, and domain Archaea, which includes archaebacteria. Domain Eubacteria includes the true bacteria. It is the largest domain that includes a large group of organisms. Eubacteria as well as archaebacteria are prokaryotes. Conversely, the eukarya is composed only of eukaryotes, unlike simply prokaryotic cells. Eukaryotic cells tend to be more complex, and these include unicellular as well as multicellular protists, plants, algae, and animals. So when we're talking about prokaryotic cells, we're talking about cells that do not have the nucleus, and they exist almost everywhere on Earth. They grow in numbers so great that you can be able to see them with your naked eye. So you can be able to see them with unaided eye. They are placed in either the Eubacteria or Archaebacteria kingdoms, and they make up the smaller of the two kingdoms. Eubacteria, on the other side, make up the larger of the two prokaryote kingdoms. So literally, Eubacteria means true bacteria. They include all bacteria except for Archaebacteria. Eubacteria are prokaryotic organisms. That means they lack a membrane-bound nucleus, predominantly unicellular and with DNA in a single circular chromosome. Prokaryotic eubacteria includes bacteria with cell walls consisting of a peptidoglycan. However, not all bacteria have cell walls, but all eubacteria have a cell membrane. Now, the bacteria cell membrane consists of glycerol as well as fatty acids combined together by an ester bond. Examples of eubacteria include E. coli, Staphylococcus, Salmonella, and Lactobacillus. Then we have cyanobacteria. Now, cyanobacteria are aquatic and photosynthetic. Now, that is, they live in water and they can manufacture their own food. Because they are bacteria, they are quite small and usually unicellular, though they are often grown in colonies large enough to see. They have the distinct of being the oldest known fossils, more than 3.5 billion years old. So these types of bacteria are bluish and greenish in color, and because they are photosynthetic and aquatic, they are often called blue-green algae. The name is convenient for talking about organisms in the water that make their own food, but does not reflect any relationship between the cyanobacteria and other organisms called algae. So these organisms do not contain the same type of chloroplast as plants do, as much as they contain membrane that carry out the process of photosynthesis, but they do not contain the same type of chloroplast as the plants do. Cyanobacteria are relatives of bacteria, not eukaryotes, and it is only the chloroplast in eukaryotic algae to which the cyanobacteria are related, and they can survive in extremely hot environments, also even in extremely cold environments. Then we've got archaebacteria. Now, these are unicellular organisms in the domain Archaea, which generally distinct from bacteria and eukaryotes and often inhabit extreme environmental conditions. These lack important carbohydrate found in cell walls and have different lipids in their cell membrane. Examples of archaebacteria include halophytes, which are microorganisms that may inhabit extremely salty environments. They also include methanogens, which are microorganisms that produce methane, as well as thermophiles, which are microorganisms that can thrive extremely in hot environments. They evolve separately from eubacteria as well as eukaryotes. They are similar with eubacteria in being prokaryote and lacking distinct nucleus. These bacteria may differ in terms of ribosomal structure, the possessions of introns, and in membrane structure or composition. They are similar to eukaryotes in ways that Archaea possesses genes and several metabolic pathways that are more closely related to those of eukaryotes. Notably, the enzymes involved in transcription as well as translation. Let's discuss identifying prokaryotes. How can we identify prokaryotes? So we can see prokaryotes in terms of their cell shape, the cell wall, or even the movement. So all cells share four common components. For instance, a plasma membrane, that is the outer covering that separates the cell's interior from the surrounding environment. They also share a cytoplasm, which is a jelly-like cytosol within the cell in which other cellular components are found, or rather other organelles are found. They also have a DNA. This is the genetic material of the cells. We also have ribosomes where protein synthesis occurs. However, prokaryotes differ from eukaryotic cells in several ways. How? A prokaryotic a prokaryote is a simple single-celled, that means they are unicellular organisms and they lack an organized nucleus or any other membrane-bound organelle, right? Most prokaryotes, as I have mentioned earlier, they have a peptidoglycan cell wall and have many of them have got a polysaccharide capsule. The cell wall acts as an extra layer of protection. It helps the cell to maintain its shape and also prevents dehydration. The capsule enables the cell to attach to surfaces in its environment. Some prokaryotes have flagella, villi, or ephemerae, right? Now we're going to discuss this later in this session. Now, when it comes to the shape of the cell or the shape of bacteria, bacteria are classified into different groups. They are classified according to their basic shapes. For instance, we've got a spherical shape, which are also known as cocky. We also have a rod shape, which is bacilli, and we've got a spiral shape, which is spirala. We also have a comma, or as well as a corkscrew. Now, they can exist as single cells, or they can exist in pairs, in chains, or even in clusters. So when we're talking about the cocky, which is the spherical, now we're talking about a sphere-shaped bacteria. Then the rods or the bacillus are rod-shaped like bacteria. Then we've got a spiral, which is like a spiral-shaped bacteria. Then, as I have mentioned, that we also have a flagella, which is a leg-like structures that help to propel the bacteria. Bacteria can be gram-positive or gram-negative depending upon the staining methods. Gram-positive bacteria have several layers of peptidoglycan in their rigid and thick cell wall, which is highlighted by gram staining. The cell wall of gram-positive bacteria contains of lipotecoic acid, which is composed mainly of alcohol and phosphate in the plasmic space. This acid function helps the bacterial cell to grow. It also protects the bacterial cell wall from lysis. The acid provides the cell wall antigenic specifically to gram-positive bacteria. Therefore, it helps in the grouping of gram-positive bacteria into several types. Then the gram-negative bacterial cell walls consist of only one layer of peptidoglycan and has no lipotecoic acid, so gram staining cannot bind to their cell wall. As a result of this weak structure, the gram-negative bacterial cell wall is susceptible to mechanical damage. The outer membrane of the gram-negative bacterial cell wall consists of negatively charged particles such as lipopolysaccharides, phospholipids, and lipoproteins that enable gram-negative bacteria to evade immune cell detection and the action of complements produced by the immune system of the host. Moreover, this outer structure protects gram-negative bacteria from bile salts, heavy metal, digestive enzymes, detergents, and some antibiotics such as penicillin. The cell wall of gram-positive bacteria is composed of thick layers of peptidoglycan, and the cell walls of gram-negative is composed of thin layers of peptidoglycan. In the gram staining procedure, gram-positive cells retain the purple-colored stain, and in the grain staining procedure of gram-negative, they do not retain the purple-colored stain. That means the gram-positive bacteria produce exotoxins and the gram-negative produce endotoxin. In terms of the movement of the bacteria, so how do bacteria move? We've got a tail-like structure that weaves around to propel the bacterium, which we call the flagella. We've got cilia, which are short hair-like structures that move in a beating motion that helps the cell to swim. Then we've got sticky cilia-like structures that keep the bacteria from moving. Now, these are non-motile. So these are the different structures or the different pictures that show the different structures which help with the movement of bacteria. So plants are not the only organisms that produce energy of the sun to make food, right? Now, some bacteria can also perform photosynthesis. So now we'll be discussing bacteria and their energy. So they can also perform photosynthesis. In fact, the first photosynthetic organisms on Earth were bacteria. That's what the history says. Photosynthesis is just one of many ways that bacteria can obtain energy. So we're going to discuss three ways: autotrophs, chemotrophs, as well as heterotrophs. Now, the autotrophs store chemical energy in carbohydrate food molecules they produce themselves. And now, food is chemical energy stored in organic molecules. Now, this food provides both the energy to do work and the carbon to build the organic structures from cells to organisms. Because most autotrophs transform sunlight to make or synthesize food, we call this process which they use for the synthesis. So with autotrophs such as cyanobacteria, they make their own food or their own energy using solar energy. Now, what about chemotrophs? What are chemotrophs? Now, these are organisms or bacteria, such as archaebacteria, which make their own energy using chemical energy. So, bacteria are not only autotrophs, they can also be chemotrophs. Chemosynthetic bacteria obtain energy by breaking down chemical compounds in their environment. An example of these, as I have mentioned, archaebacteria. So one of these chemicals can be broken down by nitrogen containing ammonia. These bacteria are important because they help the cycle of nitrogen through the environment for other living things to use. Nitrogen, for example, cannot be made by living organisms, so it must be continuously recycled. So these type of bacteria help in that process. So we need these organisms for that. We need this bacteria for nitrogen fixing. We're going to discuss that later in this presentation. Then we've got heterotrophs. Now, heterotrophs cannot make their own food. So they must eat or absorb food. They obtain food by eating, for instance, E. coli. For this reason, heterotrophs are also known as consumers. Consumers include all animals and fungi and many protists and bacteria as well. But for the essence of this presentation, we are discussing bacteria. So they can either consume autotrophs or other heterotrophs as well. Then coming to the respiration of bacteria respiration. Now, respiration is part of how cells make energy. Bacteria have two ways of making energy. So this is aerobic respiration, which involves oxygen, or anaerobic respiration, which doesn't involve energy. Now, let's get to it. Let's discuss it. Like other living things, bacteria respire, right? They oxidize food materials present in their cytoplasm to obtain energy. Most bacteria make use of free oxygen of the atmosphere or oxygen dissolved in the liquid environment. Now, these are called aerobacteria or aerobic bacteria. They are so called because they can live only in the presence of free oxygen. Now, this free oxygen is necessary for their respiration. So, with aerobic bacteria, they cannot live without oxygen. That's basically what we mean, right? Now, this free oxygen diffuses through the bacterial cell walls and oxidizes the food materials present in the cytoplasm. The reaction takes place in two steps, right? The first steps include oxidation of food material, with the removal of pairs of hydrogen atoms. Then the second step now comprises of oxidation of hydrogen atoms by oxygen with the liberation of energy. The released energy is captured in the adenosine triphosphate, which is the ATP, formed from the adenosine diphosphate, which is the ADP, as well as the phosphoric acid. Now, the oxidation of food thus results in the production of carbon dioxide, water, and the formation of ATP. Carbon dioxide diffuses to the exterior through the body surfaces. Right. Then we've got anaerobic or aerobic bacteria or anaerobic bacteria. Sorry for that. I'm saying we have anaerobic bacteria or anaerobes. Right. Okay. Now, these are a considerable number of bacteria which are able to live or multiply in the absence of free oxygen. So they can live without oxygen, right? In fact, they perish in the presence of free oxygen. Now, a good example of these bacteria which decompose glucose to form alcohol and carbon dioxide, right? Now, these type of, as I have mentioned, the bacteria that can live and multiply in the absence of free oxygens are called obligate anaerobes, right? Or anaerobes, right? Now, for examples, syphilis bacteria. Also, those that can live and grow whether there is oxygen which is present or not, they are called facultative anaerobes, right? So we've got 3 types that you need to pay attention to understand, but they're quite easy to understand. One can live without oxygen. The others, they can live whether there is oxygen or there is no oxygen present. Then the other types of bacteria, they cannot live without oxygen. Then let's get into the reproduction of bacteria. Then we're going to discuss 3 ways. We're going to discuss binary fission. We're going to discuss conjugation, then spore formation. Now, with binary fusion, bacteria reproduce through this process called binary fusion. What happens now? What happens in this process? During this process, the chromosome copies itself, forming 2 genetically identical copies. Then what happened after that? Then the cell now will therefore enlarge and divide into 2 new daughter cells, right? As you can see in the picture below. Now, these 2 daughter cells, which are identical to the parent cell, right? Now, the binary fission can happen rapidly, right? Some species of bacteria can double their population in less than 10 minutes. So this process makes it possible for a tremendous bacterial colony to start from a single cell. Now, then we've got conjugation. Now, in this process, the DNA passes through an extension of the surface of one bacteria and travels to another bacteria, as you can see in this diagram. Then bacteria will therefore exchange DNA via conjugation. Then we've got, now this is a type of sexual reproduction, right? So then what will happen? We also have spore formation, right? This is a type of dormant cell which exhibits no signs of life. It is highly resistant to environmental stresses such as high temperatures, irradiation, strong acids, disinfectants, right? So the endospores are formed by vegetative cells in response to environmental signals that indicate a limiting factor for vegetative growth. Now, let's discuss symbiosis. Now, a close relationship between two species in which at least one species benefit from the other, right? Now, symbiosis is any type of a close or long-term biological interaction. So now these are the relationships between two different biological organisms, be it's mutualistic, commensalistic, or parasitic. Right. Now, with parasitic relationships or parasitism, bacteria exploit now the host cell, injuring them, causing diseases. For instance, the bacteria, which is Mycobacterium tuberculosis, which causes TB. Then we've got a mutualistic relationship now where both the host and the other organism benefit. Both organisms benefit in this way. Now, in this type of symbiosis, these two species live together in such a way that they both benefit. For instance, E. coli. Then the last one, the last discussion for today or for this session, symbiotic bacteria are bacteria living in symbiosis with another organisms or with each other. As I have mentioned, for example, the rhizobia living in root nodules of legumes which provide nitrogen fixing activity for the plants, right? Now, when we're talking about nitrogen fixation, we're talking about a chemical process by which molecular nitrogen with a strong triple covalent bond in the air is converted into ammonia. So it is very important, as I have mentioned earlier, that as human beings, we cannot make nitrogen. Therefore, bacteria play a major role in fixing nitrogen so that it can be consumed from consumers, and then it ends up being decomposed and then sent back to the soil. Now, nitrogen, atmospheric nitrogen is molecular dinitrogen, a relatively non-reactive molecule that is metabolically useless to all few microorganisms. So now, when we're talking about nitrogen fixation, this is essential to life. Why? Because fixed inorganic compounds or nitrogen compounds are required for their biosynthesis of all nitrogen containing organic compounds such as amino acids, proteins, nucleosides, triphosphates, nucleic acids as well. So as the part of nitrogen cycle, it is essential for, let's say, for the manufacture of fertilizers or in agriculture in general. Now, nitrogen cycle or nitrogen fixation rather is carried out naturally in the soil by microorganisms termed diazotrophs that include bacteria. So it is important for us to acknowledge bacteria, because without these nitrifying bacteria as well as denitrifying bacteria, which help now to convert this nitrogen that was in the soil already back into the atmosphere, then we wouldn't have nitrogen. So yes, some of these nitrogen fixing bacteria have symbiotic relationship with plant groups, especially with legumes. So this is more of a mutualism or mutualistic relationship. That is for now. Thank you so much for joining me. I hope you enjoyed this lesson. Thank you so much. Until next time.